Thank you, Dr. Narang. Next up, the bioengineering industry. It's exciting and full of potential. But what lies ahead in the future of bioengineering? Passing the time over to you, Dr. Palmer.
Hi, I'm Megan Palmer. I'm the Executive Director of Biopolicy and Leadership Initiatives at Stanford University and an adjunct professor in the Department of Bioengineering, as well as an affiliate of the Center for International Security and Cooperation. Today, I'm going to tell you about our work on engineering biology at social scales. I'm a bioengineer by background, but for the last 10 years or so, I've been working on the social, organizational, and policy issues coupled to advances in synthetic biology and bioengineering, and hope to share some of the work that's going on in this field, and in particular, some of the work that has been developed through the International Genetically Engineered Machine Competition, or IGEM, which is shown here. So to begin, I know a lot of the discussions during this conference are about technology, and I would like to start with the premise that really all of technology is biology, right? Biology is a technology that has grown to take over the world even without us, and everything that is human mediated in terms of the technologies that we build is in some ways part of our living systems. And we are inspired by biological systems. We're inspired in the things we build, and we're inspired in part because we are made of living matter ourselves. But today I want to focus in particular on how in the field of synthetic biology and engineering biology, we've been thinking about biology as technology. Biology is a really amazing technology because it is able to take in very simple ingredients like air and sunlight and water and sugar and produce a vast array of molecular compounds, as well as a vast array of behaviors, forms, and functions. And this technology is also able to be encoded through the use of the genetic code here, the A, C's, T's, and G's that form the blueprint for these types of molecular functions and forms. And astoundingly, it can replicate itself. So it again grows without our control. But increasingly, the scientists and engineers that are part of a number of fields, but notably in bioengineering and in synthetic biology, began to want to engineer these types of living systems with the same type of precision and control and modularity that we see in other types of systems and with other types of substrates. So what would it mean if you could engineer living systems at the level of genetic code and then also take advantage of this additional engineering toolkit through reproduction and evolution of these systems? And so the fields of engineering biology and synthetic biology brought in inspiration from many different science and engineering disciplines, from computer science, electrical engineering, mechanical engineering, and more. And this has really developed into its own field of science and engineering. And over this period of time, we've developed a pretty amazing engineering toolkit. Many of you know about the Human Genome Project in demonstrating the ability to read the genome, read all of the molecular blueprints on which we are built. Increasingly, it's becoming easier to write this genetic code through DNA synthesis. We also have toolkits in being able to edit and evolve these systems. So editing has been a particular topic of interest with CRISPR technologies, but this is a much longer history of recombinant DNA technologies, over 45 years. And also the ability to evolve these systems, putting microbes and other organisms into environments in which we can direct their evolution towards certain types of traits. And it's really this combination of all of these different tools that is enabling this field, and in particular allowing us to move between a digital space and a physical space. So we're not only working in a lab with these physical materials, but we're able to engineer and design organisms and functions in digital spaces. And so this is just one example. This is an image from a company called Ginkgo Bioworks that I'll mention later, in which the modern bioengineer is sitting on their computer designing a organism, or really a whole set of organisms that robots around them are able to help design, build, and test. And it's this iterative cycle using all of these tools that is able to produce all different types of valuable things. So what type of valuable things are we talking about here? So on the top of most of our minds is the ability to use biotechnology and engineering biology products to produce therapies and countermeasures and new drugs to diseases, as well as to understand those diseases. And so COVID-19 and the development of next generation vaccine platforms, as we're seeing today, as well as the testing and diagnostics, is a very powerful application of these technologies. But there's also a number of other technologies that are building on foundations from the past as well in agriculture, and developing the next generation of plants to able to be resistant to drought, as well as engineering the supportive environment of those plants, like engineering the microbes and other environments so that they can directly, for instance, fertilize those plants without the use of fertilizers that can degrade the environment. We also see, a big part of biology has been the consumption of animal products, and now we're seeing replacements for these. This is actually not an animal-based burger, it's a plant-based burger depicted here, in which we're able to take the particular genes that encode the heme protein that looks like blood, but put it in a fully plant-based product that's more sustainable. We also see the development of biologically based manufacturing for new types of textiles and materials. So what looks like leather here is actually made from mushrooms and mycelium. And on the bottom, these are images actually of spider silk made without the spiders, made from bacteria that are encoded with those same genes and can produce them in much more scalable ways. We also see elements like concrete and other types of building materials being able to be developed using biomanufacturing platforms. And then there's other types of non-intuitive applications, like the development of data storage using the genetic materials, right? And it's actually a very robust way in which to store that type of information in the long term. There's some new projects that have been advancing the forefront of that field. So a vast array of different applications across many sectors. Biology really can touch almost everything that we use in our daily lives. And it's been notoriously difficult to estimate the scale of the bioeconomy, but this is one example from McKinsey last year, estimating the impact of this bio-revolution, noting that biotechnology is only sort of one element of larger bioeconomies, but estimating it at values of 2 to 4 trillion in 2030 to 2040. So I have spoken at a high level about the types of technological transformations and transformations at the molecular scale. But what's critical to understand in this field is it's been enabled by and accompanying transformation at the social scale. And one example of this is the iGEM competition. This is a competition that actually started as a course at MIT in 2003, right about when I was beginning my PhD in bioengineering there. And the idea was fairly simple, that the scientists and engineers in this class wanted to design simple organisms using using simple genetic parts. And this idea has actually grown over time in the last 15 years. This was the competition last year. Also still remained in Boston, but had to find a much bigger space than MIT. That gathered 360 teams from I believe 4 dozen countries to participate in this yearly competition to design living organisms. You can see some of the places where these teams are from. They're not everywhere in the world, but there are many places around the globe. And the basis of this competition was a scalable toolkit. The idea of each bit in this kit of genetic parts are tiny pieces of DNA that are made by teams and contributed to a growing repository of tools that teams can mix and match to build their living organisms. And the magic in this competition is that teams don't build an organism to win a game like in other competitions, but rather to do the most useful thing that they can imagine using bioengineering. And over time, this has grown to a wide array of the applications, like what I highlighted before, from manufacturing to health to food and energy to foundational tools. And increasingly, teams are inspired to be mapping these particular problem sets to larger global problems of which biotechnology and technology can play an important part, like the Sustainable Development Goals. And this year at the competition, there was actually a prize specifically around the Sustainable Development Goals, and the whole competition went on virtually. Now, just to give a glimpse at what these projects look like, this was an early project. It was actually a winning project from Cambridge, in which teams wanted to build upon the parts for sensing in the environment and tuning control to that to a useful readout, in this case, a colorimetric readout of what was occurring in a cell. And they developed pigments that could be genetically encoded. And then they worked with designers to imagine potential products that would be built, in this case, a probiotic that when ingested could sense what was going on in the gut and develop a useful readout of what might be going on. And teams then share these results through online wikis, which essentially lab notebooks, as well as posters, presentations, and then they contribute their new genetic parts to this growing repository. And they get prizes. They get medals non-competitively for achieving certain feats, awards for being best in class, and the overall winner gets a bio brick trophy, a big Lego brick. And they're adjudicated by near peers, people across sectors who come to this competition every year to evaluate the results. And you can see some of the examples here from a few years ago of teams interacting, not just with these judges, but really with other teams as well to share their results. And at the end of the day, they get to present to this global community on their innovations. And it's really outstanding. It's an amazing event to participate in. And over time, we now have thousands of projects, over 3,000 projects across all of these different areas. It shows the number of teams in these different tracks that I mentioned before. And each of them is essentially a prototype for the things that can be built using bioengineering and synthetic biology. And many of them have formed companies over time to continue to pursue that past their one-year projects. And this is just an image of some of the teams that have participated. And this is what I think really brings people back, are communities of global innovators working on some of these difficult challenges together in a collaborative as well as competitive environment. And it has grown to be quite an enthusiastic community over time. So, what are the implications of this field? There's a lot of stories that you'll hear on newspaper headlines, on magazine covers like this, that presents these advances in bioengineering as often simultaneously saving and destroying the world. There's much promise and much peril. And as asked in this foreign affairs article, should we be happy or should we worry? And I'd like to argue that it's probably both, and we need to be both happy and optimistic and promoting this technology while also doing so very, very carefully. And these are some of the reasons why we need to be attending to these issues with particular care. As one example, these new types of tools are enabling us to do potentially very powerful new things. So this is one example illustrating the use of a gene drive. And gene drives actually leverage some of these CRISPR technologies in order to enable the passing down of traits through populations in greater efficiencies than you would see in systems without the drive. And what that enables you is to drive a trait through a whole population of a species. In this case, imagining things like either eliminating a potential mosquito that could carry a malaria, right? And in this case, it's potentially solving one of the biggest global health challenges, and yet the capacity to introduce one organism and changing things on global scales across boundaries needs to be carefully considered. There's other types of applications like solving supply chain challenges through the development of other biomanufacturing platforms for both existing and novel drugs. But what does it mean, for instance, if you produce a production platform for opiates that both creates new possibilities for acquiring such capabilities, but also disrupts supply chains that have geopolitics accompanying? There's also been some conversations about the capacity to engineer ourselves. What does it mean, and should we be using these technologies to edit our own genomes, especially when they impact not only us, but future generations? And one that is certainly top of mind now is that each of these technologies is inherently dual use, and meaning that it can be used for helpful purposes, but also for harmful purposes. And so how do we ensure that these technologies are not developed for harm? These conversations are actually quite old in many ways, even though the modern field of recombinant DNA technologies is only 4 dozen years old. This is actually 1975 here at the Asilomar conference, where scientists from around the world gathered to discuss what the rules of the road should be around their new technologies. And in many ways, we're using these same forums today. On the right is the Human Genome Editing Conference, gathering in many ways some of the same people. And it's not only in scientific communities, we also see discussions at the United Nations and elsewhere with countries around the world, making important decisions and reinforcing important decisions. For instance, banning the use of biology as a weapon. That's such an important foundation that we need to recommit to in order to take advantage of the benefits of these technologies for peace and prosperity. But I'd like to argue that this same competition, this iGEM competition, has been an important next generation of conversation around the technology and policy space in biotechnology that allows an ongoing, grounded observatory into the frontiers of science and innovation and how we need to adjust both our technologies and our policies and the coupled practices in this field. In the context of the iGEM competition, which I mentioned before, this has taken the form of a number of programs that have grown over time. One of them is the Human Practices Program, which I've directed at iGEM for the last 10 or so years in collaboration with many other volunteers. I'm a volunteer as well as the Safety and Security Program that stemmed out of this. And this is actually Pierce Millett, who heads that program. And he used to be staff at the Biological Weapons Convention Implementation Support Unit, now helping to safeguard the frontiers of the field. And what we do through the competition is actually ask each of the participants to demonstrate to us that they have thought carefully and creatively about whether or not their work is responsible and good for the world. And by responsible, we mean conducted with care and foresight. And good for the world means that they've been able to provide evidence that their projects can be reasonably anticipated to benefit and not harm people, to distribute benefits fairly, and other types of ethical considerations that are coupled to this work. And one of the things we've had to realize is that none of these questions have single or simple answers, and there's a diversity of approaches that we we need to take, and we need to do so collaboratively, not ignoring many of the things we know in the past, but building upon them and finding the gaps and doing an iterative process of finding out the policy puzzles and working on them in conjunction with technology development. This is actually embedded within the award structure for the competition. So teams can't participate unless they comply with an evolving set of policies that are adapted every year. But they also have to show that they've justified why is their project useful. They need to show that they've considered safety and responsibility. And then they need to show us the ways in which they've addressed those issues within their project, either through the technologies that they develop, the goals they pursue, or the types of accompanied policy and organizational practices. And then we give prizes for those that excel and innovate. Over time, groups of volunteers have provided guidance, examples of exemplary projects, talked about the history, and there's a committee that gathers every year to adapt these rules and observe what occurs. We also have an increasingly state-of-the-art, sophisticated global biosafety and biosecurity program that involves expert advisors from all around the globe, an evolving set of policies, as well as guidance to teams, and incentives. There are prizes and awards given out in this area as well. And then we screen that whole collection of parts through partnerships with a variety of groups and have expert advisors who work collaboratively with teams to identify early and often the types of issues that might crop up in their projects. And what this has led to is a really amazing set of examples of engagement of teams that are working with their local communities on both local problems and global scale solutions. We've seen collaborations with between teams and companies to identify gaps in policies, but importantly, fix them. Working with law enforcement as well. We've seen tools around greater accessibility. We've seen the development and engagement of new programs around funding and oversight within countries that didn't have any bioengineering programs at all. And so one of the big questions is, we see these anecdotes, but how do we really know that this has had an impact both in the world and in the lab? I'd argue that one of the most important things is just to see what people do in later stages of their careers. So the first competition participants was a team from MIT. They're some of my colleagues from back then and classmates. And they've actually now formed a company that does the same thing as the competition. They design organisms, custom organisms for many applications, now valued at more than $4 billion. And importantly, these lessons around working on some of the most important problems and being able to rapidly integrate many considerations and pivot have borne out in practice with leveraging their platform to develop next generation diagnostics, therapeutics, and vaccines. We also see this again feeding back into an ecosystem, inspiring other teams. So this was the winner in the one of the winners this year. The competition was a team that actually worked on diagnostics around current and future pathogenic threats. And these teams are assuming leadership roles, not just in technology development, but also policies. So some of the folks here were in the delegates program for the competition and now are working for organizations shaping policy at really a global scale, both IGEM itself, as well as some of the partner organizations that work with the UN and the WHO and elsewhere. My group at Stanford, collaboration with other groups like Mark Santolini here at the Center for Research and Interdisciplinarity, have also started a major new program to learn systematically across the entire history of the competition in a data-driven approach to learn from all of these communities and see what they do throughout their careers, both in innovation and in promoting responsibility in practice. And this type of approach has also informed other groups. So this is a group that I now chair with the World Economic Forum, co-chair with the World Economic Forum on synthetic biology, that's looking at how we develop the global bioeconomy and synthetic biology community in ways that are more equitable and sustainable and resilient. So we didn't have a lot of time today, but hopefully I've inspired you to think about biology as a technology and technology as biology, and realize that this is something to pay attention to in concert with all of the changes in the world, because it's a technology that works, and engineering living matter is something that will matter in the rest of our lives and beyond. Thank you so much, and I hope you enjoy the rest of the conference.